My name is J.C. Gumbarts. I'm a assistant professor here in the physics department at Georgia Tech. And it's my great pleasure today to introduce to you Dr. Alec Aksimentiev, professor of physics from the University of Illinois at Urbana-Champaign. So like many of us in biophysics, Alec took a winding path there. So he started out, he did his master's degree in particle physics in his native Ukraine in 1996, I think. After that, he did his PhD in chemistry at the Institute of Physical Chemistry in Warsaw in Poland. After that, he did a couple of years in Tokyo at a material science institute, after which he came to Illinois as a postdoc. And that's where I first met him, in the theoretical and computational biophysics group, where I was a graduate student from 2001 to 2005. And in 2005, he became a professor, an assistant professor at Illinois. So where he was a postdoc, then he effectively was promoted to a professor of physics, where he has been since. And now he's a full professor there. Just a little personal note, I'm very proud to say that after he became a professor, I got his office space. So I got to sit at his desk. And that desk has now produced a number of professors. So there's maybe something magical about it. I don't know. So now Alec works in the field of biophysics, but more specifically at the interface of biology and technology. So I think this is particularly appropriate for us here at Georgia Tech. And so today he's going to tell us about his work in this area related to sequencing of DNA. Okay, and so with that, thank you.
Thank you very much, JC, for the very kind introduction. Actually, first time for me to be in Atlanta, Georgia. Apparently, it's really wonderful weather here all the time, like today. Is there feedback? Should I move it? All right. So if you can hear me, okay. So I'll begin. So as JC said, I had a winding path to where I am now, and he also told me just a couple of minutes ago that most of you have some sort of physics background, which makes it easier. So I come from the Department of Physics, and the questions that many of us physicists eventually come to ask is, what is life, right? So how is it possible that a collection of proteins, DNA, lipids all come together and form a biological cell? So it's a profound intellectual question, and perhaps the most important question in the field of biophysics. And of course, once we know what life is, what makes life, we could talk about treatments and drug development and so on. But as a physicist, I am always looking forward to find a simple solution to such a question of what life is. And the best solution for a physicist, of course, is a formula, right? F equals ma, or E mc squared, and so on. So for biophysics, unfortunately, there isn't a simple formula that describes pretty much anything interesting, with one exception. And that exception is the molecule of DNA. As many of you know, DNA is the carrier of genetic information, and frequently we think about DNA as this long string of letters, A, C, G, T. All of them make up the genetic code. So in a way, that string of letters is a formula, because it contains all instructions necessary to manufacture a living thing. So DNA is found in our cells inside the nucleus, and at some stages of a cell development, it can be visible in light microscope. So that's where chromosomes comes about. So as you're probably all aware, we have 23 chromosomes. Actually, 2 pairs of 22 chromosomes, one from a father, another from a mother, and 2 sex chromosomes. So if you take them out of a cell and stain with a certain dye, you can, well, maybe not you, but a professional can easily tell which chromosome it is, just by the way, what kind of patterns they form. So again, here you're looking at the scale of, tens of micrometers. So what is important to realize that those chromosomes, it's not just DNA, it's actually very densely packed fiber made of proteins, DNA, and some other things. And, but there's only one DNA molecule in it. Very long, one continuous DNA molecule that is wrapped around multiple times at multiple levels into chromosomes. Of course, during different stages of cell life, these chromosomes they can dissolve and then they can reassemble. So typically, chromosomes come to the compact form when the cell is about to divide. So the DNA molecule that is lining threads that is threaded through this chromosome is what carries the genetic information. So let's take a close look at the molecule. So this is the building block of the DNA, so called the DNA nucleotide. And that's where the sequence is written in. So there are several parts that one can identify. There is a base, and I will frequently interchangeably use nucleotide and a base. And those are the parts of the nucleotides that are different among different letters, A, C, G, and T. There's a common element called sugar and the phosphate. So the phosphate is something that gives DNA a negative charge. So a single molecule of DNA is a polymer made of many nucleotides that are arranged sequentially. So in this particular conformation, you look at one nucleotide that is highlighted, but we can kind of turn on the color for all other nucleotides. And that's basically where the sequence of DNA is. So you see, there's a small differences, just a few atoms between different bases, and that's what makes the genetic alphabet. So an important point from this slide is that the DNA code is written in atoms. So that's ultimate nanotechnology. There's nothing, you cannot physically come up with anything smaller than that, unless you go to quarks or subatomic particles. This is the ultimate density of information, and it is in our bodies. To make a double-strand DNA from a single strand, you add the complementary strands, and I'm sure you're all familiar with the story. The important information here is, again, the size. So the DNA molecule is about 2 nanometers in diameter. So the size of individual nucleotide is about 3 angstroms. And yet, the nucleus can somehow pack these molecules within a sphere of about 10 micron radius. So at first look, this cannot happen. It's not only that it's so long, it's also highly charged. So each nucleotide carries a charge of 1 electron. So you have 1 billion nucleotides, so you have very, very charged system. So how is it possible? So actually one number that people frequently quote is that the size of your DNA is about 2 meters, and that's true. All right, so some basic physics, same charges, of course, repel. That's what the textbook tells us. But it's not always the case. It's only true in vacuum. If you place 2 charges in an environment, the environment can produce effective attraction between 2 charges. And this is really important to understand how DNA is packed inside cells. DNA is a highly charged entity, but because life began in the ocean, salty ocean, it is surrounded by salt. Just like actually it's interesting to think that the environment of our cells, what's inside, actually reminds a little bit of what probably the ancient ocean was. It has a lot of sodium. It has also potassium, magnesium, calcium, and other multivalent ions. So the interesting fact that comes from physics is that the DNA molecules, they they are known to repel one another when they are surrounded by ions, simple ions, such as sodium. But once, if we replace all of the ions with those that carry higher charge, this effective interaction can become attractive. So two DNA molecules can attract one another. So let me give you a very simple, and it's designed actually for high school students, example, how is it possible, how two DNA molecules can attract one another. So first, let me do a demonstration. So you're looking at a result of a molecular dynamics simulation. This is kind of a computational method that we use. And I'm not going to explain how it works. I'm just going to show you the result. So we have two DNA molecules, and they are placed in a box of water with salt. So the spheres here are the salt ions. Water is not shown. So you're going to look at two DNA molecules interacting with one another. So one is placed in a solution of basically cooking salt, and the other one is salt that is used sometimes on the highways. I don't know if you get snow here or not, but in Illinois we do. And one thing you can strikingly see from this simple simulation is that the DNA molecules in so-called monovalent electrolyte, they effectively repel one another, but in magnesium they kind of come together. Right? They don't stick together, but they do not repel really vigorously. So what's going on? So the simple picture here is the following. So when you have two DNA molecules in vacuum, both carry negative charge, they both repel one another. Now, if we add a little bit of salt, so think it's like minus plus, it will ideally compensate the charge of the DNA. And so will happen to the other one. So now if we combine the two together, their effective charge will be close to zero, but because the compensation is not ideal, you always have some remainder of a charge. So they will push one another away. But if you take something that has a charge of +2 or higher, so it's like having something that is able to do a double handshake, right? So you would half of a charge would interact with one piece of DNA, and another half of a charge would interact with the other. So that's kind of an intuitive picture to have in mind. In fact, if we look at the simulation of DNA interaction, we'll see this double handshake mechanism explicitly if we look how, for example, magnesium ions is interacting with two DNA molecules. So I'll come back to this in a couple of slides. All right, so when we talk about DNA, the first thing that comes to mind is the DNA sequence. And it's true. The DNA sequence defines biology, is the most important information. We take the sequence, the molecule called RNA polymerase will make a complementary RNA strand. So this is basic biology reminder for you. So here it is, this yellow thing. So that strand.
Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid
and that's
is
basically
read off.
the machine that makes the RNA.
To 3 corresponding letters on the transfer molecule.
So that's basically how we went up with the proteins. Sorry about the sound. But basically you go from DNA to RNA to proteins. And now the question is, so how is it possible that all our cells, although they carry the same DNA, they all look different? So for example, my hair cell is different from my skin cell and different from my liver cell. Like, what's going on? They do have the same DNA, we know it for a fact. So that is something, so basically the DNA sequence is not the entire picture. There's something else that's going on. And that is called epigenetics. Right? So epigenetics, so I'm going to explain to you within the next slides what epigenetics is and what it does. So to understand the mechanism of epigenetic regulations, let's take a look at the actual structure of the DNA inside the cell. So at the very basic level, we have our DNA double helix. That's where the letters come in. At the very highest scale, we will have our chromosomes. So, but in between, there's this multiple orders of magnitude complexity in arrangements of the matter. So the one scale up from the double helix is so-called nucleosome. which is basically it's like a spool, right? So we have a protein core called a histone, and then the DNA is wrapped around almost 2 turns, and so on. So it's 2 turns here, 2 turns there, and so on. So this part, we don't know actually what happens in there. And so the epigenetics happens at the level of the histones. So I'm going to explain to you what it is. To do it, let's take a look at the nucleosome particle. So again, those who are not colorblind can easily see that this is a DNA molecule wrapped around protein. I'm a little bit colorblind, so I always amazed that people can see something in here. So we have a highly negatively charged DNA. The core particle is somewhat positively charged, but not enough to compensate the charge of the DNA. But the protein has very funny structure. It has this tails. It's called tails. Those are just flexible polymers made of unstructured proteins, and they are positively charged. So each of those K, that's abbreviation for a lysine, is a positively charged amino acid. So here's the chemical structure. So again, we are back to the level of atoms. Another important player are the nucleotides. So this is a cytosine. It's one of the letters. That's the C letter, right? So far, everything is conventional. Now, where is the epigenetics? So here it is. Oh. Do you see the difference? So the difference is really tiny. So if you look at the lysine, it has a charge of +1. Now it's modified. It's methylated. It has 3 methyl group added to it. It still has a charge +1, but it's just slightly bigger. Same thing happens with the cytosine. If you look closely, this is a conventional cytosine, one of the letters. And this is a methylated cytosine. It has 1 methyl group added to it. That's it. So now imagine how is it possible that we change just a little bit in the atomic structure of a little component, which is not even nanometer in scale, and we can regulate the morphology of a cell, what a cell does. So that's a profound question. How is it possible? What is the physical mechanism for such regulation? And I'm going to show you our recent work in this area that suggests one such mechanism. So let's take 2 nucleosome particles. So every physicist would like to know what are the forces, like how do they interact? Do they like to be together, or do they hate one another and like to fly away? Yeah? So which is schematically shown here. So why is it important? Well, imagine that it happened to be that a piece of a DNA or chromatin has a landing spot here. So the DNA polymerase can go find it, and then it can do its business. It can transcribe RNA and make proteins. But if chromatin is compact, meaning the nucleosomes, they really like to stick together, well then, tough luck, the gene will not be transcribed. So this is one of the mechanisms. Is it true? How is it possible? So this is why we use a supercomputer at the University of Illinois called Blue Waters to measure directly the forces between DNA molecules and between nucleosome particles. So what you're looking at here is a movie showing one such simulation. You have 2 DNA molecules. They are connected by a spring, and they are free to move around the simulation system, but we know by how much the spring extends once they move about the system. So we directly know the force between 2 DNA molecules, and we can make the graph of the force versus distance. So you can see when 2 molecules come very close to one another, the force becomes repulsive because they just clash. It's a steric clash, but indeed, far away, they attract. And for those of you who've taken physics 212, I believe, we can also go from physics from force to energy. and draw the energy of such interactions. So if you're not a physicist, don't be turned away. So think about it like a graph that explains the interaction between the two molecules. It's like a well. If there is a well, the interaction is attractive, and the depth of the well will tell you how strongly they attract. All right, so we performed these simulations on a system containing DNA of different sequences. So just to check, we were wondering, so what will happen? So here's a movie again illustrating two molecules interacting with one another, and they are made entirely of C and G nucleotides. As previously, we see that they indeed attract, but they attract a lot less than identical molecule made entirely of A and T nucleotides. So that was really unexpected discovery that we made using a supercomputer. We found that the sequence of the DNA, or to be more precise, the content, the AT content, modulates the effective interactions between DNA. Why is this important? Well, if you look at the structure or the sequence of a chromosome, you will find that the chromosome, or the DNA in the chromosome, has islands that are rich of A nucleotides. And that is something that has no information about genes, so it wasn't clear like what exactly that DNA does. It's something that is frequently referred to as junk DNA. But our hypothesis is that it's not a junk DNA, it's a structural DNA. This is the way you fold chromatin, because AT-rich components will attract and fold this precise three-dimensional structure. All right, and that would be a very nice computational study, if not for the fact that we had a world-class single-molecule experimentalist at Illinois, TJ Ha. So they decided to test whether our predictions were correct, and the way they do is really ingenious. So so consider that. So what they do is the following: they have a slide. So that's a slide of a microscope. Well, think about glass. They are able to capture just two molecules of DNA in a lipid bubble. They have a special extruder. It's a complicated process, and it's a statistical process. So sometimes you get one, sometimes you get three, but on average you get just two molecules. And each of the molecules would have a different dye attached to it. So when the two molecules come together, a process called fret happens, which is basically this spike. So the energy in the green channel is transformed into the energy in the energy in the red channel is transformed into the energy in the green channel. So the spike occurs, and that is the measurements how frequently two DNA molecules come close together. So they were able to do these measurements for DNA of different sequences, and to our delight, indeed, they found that AT-rich DNA attract a lot more than CG-rich DNA. So we have experimental validation for our predictions. All right, what does it have to do with epigenetics? So let's repeat the same simulations, now using DNA that is methylated. Again, remind you, methylation is just addition of those 4 atoms to each cytosine. And in this case, we again observe a much stronger attraction between DNA molecules. So all of that is consistent with what we know about the structure of chromosomes and how methylation affects it. So once the chromosome is methylated, it becomes more compact or inactive. Single molecule experiment basically confirmed what we've just predicted. Methylation indeed produces attractions between DNA. So, so what is the mechanism? So I've just given you this observation from a simulation and also from experiment, but what is the underlying mechanism? So now recall this double handshake, remember? So for 2 DNA molecules to attract one another, they have to have an ion in between to form a bridge. And that's exactly what happens here. When we have a CG type of a DNA, it has enough room for those ions to go inside of the duplex, and they do not share ions in between, so they do not attract. But in the case of A nucleotides, they have those methyl groups. So this is actually part of a T nucleotide. It also has a methyl group. So they attract by sharing the same positive particle. And same thing happens with methylated strands. So it's sharing by attraction, or attraction by sharing. Very interesting concept. So here's the overall pictures that come out from this study. So imagine this is a nucleus, and we have a chromosome in it. This is a DNA polymerase. Remember this little guy that reads the sequence of the DNA? So it comes around, and if it encounters, a methylated clusters, it can't do anything. It can't start the transcription. Same thing happens if it encounters the structural DNA. But once it finds an open conformation, it can transcribe the messenger RNA. The messenger RNA will be transcribed into the protein. So this is one example where we used physical methods to suggest a possible mechanism of regulation, of epigenetic regulation. All right, so let's go back to the sequence. I said that the sequence is very important, but it's kind of an old news. You probably know that the DNA sequence has been already found many times, right? The Human Genome Project finished in 2003, was super successful. It was one of a few projects that actually finished before it was planned to finish, and they had surplus of money left. It's almost unheard of. So yeah, so the goal of the project was to determine the complete sequence of the DNA, all 3 billion base pairs, and here they are. Yeah, well, this is another point to illustrate that it's really long. It would take me, seriously, like 1 month just to click. It's 1 million pages. It's a lot of data. And you know, if you're a data scientist, that's probably where most of the data will be. Genomes is exploding. Well, so how big is the genome? The genome would fit on a CD. You can do the math, right? So it's one letter is 1 of 4, so it's 2 bits. It's about 715 megabases. So what's in the DNA? Like, what are the differences? And this is something that people frequently ask. You know, how is, say, my DNA is different from yours? It's actually not all that different. So for unrelated individuals, 99.4% we are identical. And just for reference, with chimpanzee it's 99%. So it's really not all that much. So I'm going to illustrate this for you. This is mine, and this is GC's. You see, there's this one C there. That's the difference. But seriously, but there's still a lot of difference. It's 1 million difference between us. And the DNA sequence is the beginning of the personalized medicine. The first step is to determine what you've got, right? Reading the sequence of DNA is essential to determine predisposition to many diseases, but perhaps the biggest impact in emerging technologies to sequence DNA will be in the area of cancer research, because cancer is a disease of DNA, and to make the ultimate diagnosis, you just have to sequence the DNA. And with cancer, it's very tricky because cancer progresses, so you need to do sequencing. You cannot do it once a lifetime. Unfortunately, you have to do biopsies. You have to take from multiple tumors and so on. So really, I think the key to solving cancer problem would be to have a really cheap and efficient DNA sequencing technology, and that's something that I'm going to tell you within the next couple of minutes. So human genome was done, right? And good. Why don't we stop there? So you have to realize that how you did the human genome. We actually did not read the sequence of the DNA. Collectively, we didn't take a book, we didn't read it, we didn't look at it in the microscope. What we did, we tricked nature to do the job for us. So this is a little enzyme. Again, it's a biological motor. Its function in nature is to read the sequence of DNA and synthesize a complementary strand, right? So that enzyme knows how to read DNA sequence, and we know how to trick it so that it will stop reading, incorporate a fluorescently labeled nucleotide, and then we can, through some tricks, put together the sequence. This is not a direct reading of the DNA sequence. And that's something that we've been working for some time. So my involvement began in 2003, I guess, when we decided, well, why don't we build basically a transistor with a hole, yeah? And read the sequence of the DNA directly, without using any kind of enzymes, just by physical measurement. So that's the ultimate goal, to have a device that you just feed DNA and read out the sequence. And it used to be a science fiction back in 2003. We are very close to having it working. But before I show you the progress, please realize how difficult the task is. So first of all, one nucleotide is just 1 nanometer. But that's not the difference. The difference is smaller, is probably 3 angstroms, just 3 atoms misplaced. Same with epigenetic modifications, just 4 atoms. And of course, we are all familiar that physics instrumentation, and physicists are very good on doing this measurements. Single electron transistor has been put together, everything been done a long time ago. But everything was at cryogenic temperatures in vacuum. Here we have DNA in solution, in water. Not the favorite environment, so it's challenging, but it works. Okay, so this is the plot that I like to show to get a perspective on how fast DNA sequencing technologies are evolving. The cost of human genome sequencing used to be $10 per base, so that was in the 90s when the human genome started. So $10 per base, it's like astronomical. It's almost mission to Mars to sequence human genome. But then it was basically dropping exponentially. As people figured out how to make devices smaller, microfluidics came on board. So this kind of improvements followed very nicely the Moore's law up to a point. So what do you think happened here? Did someone come up with something amazing technology, like maybe aliens came and gave us a device? No, actually something happens very strange and fantastic. National Institute of Health determined that this is exponential is not good enough. They said, well, if it keeps going like that, we get to the $1,000 genome by year 2040, and that's way too long to wait. So let's do something about it. And what they did, they put a call for revolutionary genome sequencing technologies. And the program manager Jeff Schloss, I think you don't get Nobel Prize for being a program manager, but somehow it really, in 2003, there was this meeting, and the goal was set that in 5 years, we drop this by a factor of $100,000, and in 10 years, we drop it to 100K, and that's exactly what happened. Amazing. So many companies came about. So what basically happened? People were able to take risk. Yeah. So there was this pool of money that they could use and try. There fairly crazy ideas were put forward. Probably 1 out of 10 or 20 worked. But at the end of the day, there was several technological transition happened. So new companies were formed. Many of them are obsolete by now. So we have like in a third or fourth way of sequencing. The most revolutionary one was this nanopore electrophoresis that I'm going to describe to you in great detail. So what is nanopore electrophoresis? The principle is very simple. You have a nanopore, first of all. Nanopore, as the name suggests, is a nanometer pore. There's nothing more to it. We placed it in salty water. We apply electric field across, and because DNA is negatively charged, it will find the pore by itself. It will go through it in extended form, And then the next one will come in and do the same. So as this happens, the presence of the DNA inside of this pore will block the current that flows through it. So really, in a way, we have an electrical measurement of DNA transport through nanopores. So it's kind of similar to like threading, putting a thread through the needle's eye, although the needle's eye is just 3 nanometers in diameter, and it happens 1,000 times per minute. And what's most importantly, you don't have to do anything, you know, it's in the dark. It happens without you. If we had to go and find the pore where to put it, it would never work, but the physics of the system is such that it works. That was just to get the gentleman up. So how can we use nanopores to sequence DNA? So one essential thing, obvious thing to realize, we have to make the pore very small. The pore has to be of the size of the DNA, otherwise it's not going to work. Yeah? So the DNA is about 1 nanometer in diameter, so where can we find a pore that is 1 nanometer in diameter? And one such pore is this biological membrane channel. So in biology, what it does, it goes and punches holes, and then the cell dies. But it also forms a very stable pore of the appropriate size. So what we can do, we can place this molecule in a lipid bilayer membrane. We add DNA on one side. Again, we apply electric field. So I'm going to show you a movie that illustrates this. So you see this is pluses, minuses. The DNA molecule will go by itself through this pore from one side of the membrane to the other. And as it passes, it will block the ionic current. So again, there are ions here. Well, here they are. So ions on one kind, those would be potassium or sodium, would go up through the pore, and of other kind would go down. So that's ionic current. You know, it's different than electric. Well, it is. Electric current is just not carried by electrons. It's carried by actual motion of ions. This particular movie, you can see those balls. Those are sodium and chloride ions moving up and down through the stem of this channel. So what was remarkable about this idea was that experimentally it was shown that this blockade, or the amount of current that is blocked, is sensitive enough to tell the sequence of DNA. So ideally, this is a picture from old review, you would expect to measure a trace just with your electrical measurements, where those steps will tell you the sequence. So direct electrical measurements of the DNA sequence was proposed. And people didn't believe it. For a long time, people thought that was not going to happen, although there's no physical reason for it not to happen, because if everything is right, it should work. Until finally, Jens Gandlach from University of Washington made it happen. So there were a couple tricks that he had to do to make it work. So first of all, he had to go to a different pore. So the pore that I showed you before was a no-starter because the stem was too long. So you measure signal from 12 nucleotides at the same time. So this is a pore that worked. Second, he had to add a biological motor there. But this time, not to read the sequence, but to produce a tug of war between the electric field that pulls the DNA down and the work of the motor that pulls it up, slowing down the DNA transport. So that was critical. And with this setup, basically, they got for the first time this stepwise signatures of DNA transport, which was, by the way, the first time the DNA was sequenced using nanopores. So fast forward a couple years, so we have now a company, which actually Jens is not affiliated with. It's a company called Oxford Nanopore Technologies. They have a device. It's basically a USB device for DNA sequencing. So you add DNA here. It works. Recently, the company disclosed what kind of pore they use. It's a biological pore. They use a helicase motor to slow down DNA transport. There are about 800 wells in this device, and it provides a direct readout of the DNA sequence. It also provides direct readout of the epigenetic sequence. So this is extremely important because everything we know from conventional sequencing comes from amplification. So we take DNA samples, and we use a process to get more of the DNA, and then we do something about it. This is the only way right now to get the sequence directly of the DNA strand. It's also very light, 100 grams. They're highly portable, and you can take it to remote places. So it was actually used to sequence Ebola in real time as disease was developed in Africa. So it works. Everyone's happy, but the problem is this one: 800 parallel wells. So we have 800 pores basically. So at first look, it's impressive. It's better than one. Yeah, 800 is better than one, but it's not enough to actually do human genome sequencing. So everything that this device was used for so far was sequencing viruses and bacteria, which have much shorter genomes. If you want to ever do it for clinical samples, we have to go to something that is much more scalable, much more like silicon nanotechnology. So back in 2003, already then, we were able to make silicon devices with feature sizes of the size of a DNA nucleotides. So I like to, this is an old slide, but I like to show it because this is a gate of a transistor, and you have a layer of dielectric here is just 0.7 nanometers. This is a nanopore drilled in such a membrane using highly focused electron beam. So basically, the dimensions of the pores are correct to read out the sequence, but there are problems with using silicon for it, and I don't have the time to go into it. If you're interested, please ask. But then another revolution came about, and a bunch of guys with Scotch tape and a microscope obtained a Nobel Prize for demonstrating graphene. And graphene is ideal material, in many opinions, to be the platform for DNA sequencing because it's so thin. The carbon layer of graphene is just a couple angstroms in height, ideal to isolate an individual nucleotide. So in principle, you can imagine making a layer of graphene with multiple pores, each of them reading DNA sequence. So here's an example of how this could work. So you're looking at the DNA molecules that is being pulled through the graphene nanopore. It's the same molecules you're looking from a top view and a side view. And one thing you'll find in this movie is that the DNA sticks to the graphene, but it remains mobile. And the reason why it remains mobile because graphene is so perfect, there are no defects in it. So any other place along the surface is the same, so it just keeps going. And while it's keep going, it doesn't go like irregularly. It actually proceeds in step, just like what the stepping motor of the polymerase did for the biological nanopore. If you look closely, you can see that the DNA advances in discrete steps, like this, which is ideal for sequencing. So we still don't know if it's going to work with the graphene. Probably will make it happen, but this is an active area of research. So, of course, sequencing using solid-state material is interesting, but it doesn't stop at graphene. There are various methods put forward. So plasmonic nanopore sequencing is something that we look at my lab. This one is fairly revolutionary. It's from a lab of Kawai at Osaka. It's quantum sequencing of DNA. So if this works, this would be for the first time that quantum mechanics was used for some direct advances in biological sciences. This is how it works. This principle is kind of similar. You have a DNA molecule passing through the pore, but now instead of reading the current of ions, you have electrodes and you have tunneling. So electrons now come from one electrode through the base to the other one by tunneling, right? And some preliminary data indicates that this could actually work. There's a company, there's a device. This is a device prototype about the size of a coffee machine. And actually, if you look at any major nanofab company like Intel, Sony, you name it, they probably have a nanopore project going on because that's finally leverage for the expensive and highly capable fabrication facilities that they have in the biosciences, right? So they can manufacture transistors with a 9 nanometer line thickness, so they can also do this. Okay, let me finish up here with a little perspective. So, okay, so let's say that we understand the biology well, and means have been developed to figure out what the problem is, so we can sequence DNA. What are we going to do about it? Right? So let's say we know the biology, we know the molecular cause of the disease, how are we going to address it? So typically what you do, you go to a doctor, they give you pills, right? The problem is that for most of the pills, we don't really know how they work. So there's these smart pills that we know how they work, but it's very difficult to get them to the correct place. Yeah. So first of all, they have to target only cells that they are intended to target, but that is easy, relatively speaking, to do because all cells have different surfaces, so you can design a drug that will interact with a preferred cell. But then you have to make sure that the drug doesn't get eaten by everything else that is in your body, yeah? So it doesn't degrade before it reaches the cell. And finally, it has to break through the boundary of a cell and enter the cell to do something there. So those are kind of fundamental challenges in drug design. Surprisingly, DNA could actually be a player in this field. Of course, there is. Drug design is a really broad field with many bright people working in it. But I find it surprising that DNA can be used maybe to cure DNA. And the technology that is used is the one that I will illustrate in this movie. So this was developed by Paul Rosamund. And it's called DNA Origami. So imagine that if I want to make a sculpture. Let's say I want to make a sculpture out of DNA. And for the reason obvious that will become, the sculpture will be the logo of the University of Illinois, which is a big letter I. How can I do it? So what I can do, I can take a long DNA molecule, and I can design a set of complementary DNA molecules that will bring different parts of the molecules together. So I add enough of those staples, so I will cover completely this long strand, and then I'll just let it go. In experiments, this takes actually longer, a couple hours, but at the end of the day, the system will self-assemble into prescribed shape, like the logo of University of Illinois. So that method is very powerful to produce arbitrary nanostructures. It has been used to make pretty much anything people thought of. You know, vases, buckyballs, gears, membrane channels. This is a transformer robot that can actually clap hands, and so on. But one thing that you can make is, of course, a jewelry box, or let's just call it a container, right? So this particular design comes from a lab in Denmark. So you have a box with a lid, and you load it with drugs, okay? The lid can be opened and closed, depending on the key. So the key is another DNA molecule that comes in, and if it's complementary, it will basically open it up. I hope this works like that. And although it doesn't work always, it can also close it sometimes and so on. But this is kind of really transformative technology in a way. You use DNA to make the structure, you load it with drugs, and it comes there and releases it. Actually, a group from Harvard demonstrated something even more impressive in 2012. If you want to read about it, just Google cancer-killing DNA robot, and you'll find it. It's the first hit. But basically, it's the same idea. You have a container made of DNA origami. It's loaded with drugs, but it had two additional things. It had antibodies that could recognize only cancer cell. It would stimulate the uptake of the robot, and then it would open up once it's inside. So it's kind of science fiction, but proof of principle has been done. So maybe the delivery, the next drugs would have some DNA in it. So here's something that actually comes from my work. Another way to do targeted delivery is to use a syringe, right? So if God forbid, but if you're sick, syringe is something that you would want to use to do targeted injection. So maybe we can do the same with a cell. So look, we want to deliver something to a nucleosome, the 6 cell, just like that. Boom. How can we do it? Well, we can design a DNA origami structure that will actually be just like a syringe. So here's the result of our collaborative work with Ulrich Kaiser from Cambridge, who come up with this syringe-like architecture that contains anchors that will enter into the lipid membrane and create a hole. So the size of this nano syringe is just the right size to deliver the majority of protein drugs. All right, so I'm almost done. So this is my concluding slide. So what are the main messages from this talk? So the first one, all the biology is very complex. Physics matters. Yeah, physics underlines interaction between individual molecules, and ultimately, we can use physical methods to understand biological processes. The second major point is that DNA sequencing will become commonplace routine. The cost of DNA sequencing, which is now at $1,000, will drop even more, probably to $100, maybe $10. It will be so cheap that we'll be using it everywhere. It's not just health. You know, you could go to a restaurant and figure out what you eat, you know, because you can sequence it and so on. And finally, the last point, DNA is actually amazing craft material. And as the founder of the field used to say, the applications are limited only by imagination. So with that, I'd like to conclude and thank the funding agency and people who are involved in the work that I showed today. Thank you.
We have time for a few questions, but as you probably noticed, we're having some audio-visual difficulties, so if you just raise your hand and then speak your question loudly, the speaker can repeat it so everyone can hear it. One question right here.
So for some of these DNA sequencing techniques using nanopores, what's the rate of like backtracking?
So the question was, what is the rate of backtracking for DNA sequencing techniques? Usually it doesn't happen if it's a free permeation, but that is not used for sequencing. Usually you arrest it with something. Once you arrest it, it fluctuates back and forth by a couple of nucleotides. Yeah.
We have one more question here.
So what are you doing about the charge delivery? For example, that which lines your genome, for example, they are charged. So how will these DNA delivery mechanisms function when you have a delivery through charge delivery?
Well, it's really, well, first of all, it depends what kind of charge it is. So the question was, would the DNA delivery mechanism work for charge membranes, right? Very good question, especially if the membrane is negatively charged. But as you know, it happens in water, so the charge would be screened to some degree. And one can use it to advantage. One just have to engineer more positive charge to facilitate, to make it insertion more probable. In fact, the way it would work in practice, probably there will be an antibody attached to the structure that would recognize which cell to enter, and then it will just puncture the hole. Yeah.
How did you make origami box? Like, isn't it really tiny?
The question was, how do we make origami boxes? So in practice, people use a computer to do it. It's like a computer engineering design CAD program. You design the shape you want, and then the computer will generate you the list of sequences you need to buy. It's more tricky to actually verify that it works, right? So that's why people publish papers then. But designing, very easy, you can do it. Yeah, like in one afternoon. Yeah.
Yeah,
What is a pH? Right. Yeah, it is sensitive because pH could change the protonation states of the origami, obviously. And at extreme pH, it would not work. Yeah, it's pretty obvious, unless, of course, unless you modify the DNA to be stable at specific conditions. Because, DNA is just, it's just a scaffold, right? It's like a pegboard. I think you can add whatever you want to it. Yeah. Yes. Yeah, the proteins are stable and happy. The cage is not super tight, let me put it this way, because it's just one layer, so it kind of fluctuates. So maybe it's not the optimal design. But there are new designs with multi-layers. And I mean, this is basically where the science or engineering is. How do you make a better cage? You have a proof of principle, but now you go and develop into something that works robustly. Yeah.
All right, let's thank our speaker one more time. If you have questions.
Thank you.